Hey Shane.
Hey, how are you?
Oh, so, um, am I doing one test or two? Because I want us to like go over the ones that I've done because I feel like BB is important for us to go over. So adding new ones into the mix seems like I'm still gonna do the one on Saturday, but my mom wants me to do two more and
Mm-hmm.
said that you guys spoke about it.
Yeah, I think the one that you're gonna do on Saturday could be enough. So let's, I guess, do that. And yeah, let's go over the ones that you've taken. Yeah.
Okay, all right. So I was trying to look at compound one to see if I could recognize any.
Mm-hmm.
I see sulfur groups. I'm not sure where the amino acids are because
So what?
it
So
seems
what
too
does the
close.
peptide bond look like?
A peptide bond?
Yeah.
Isn't that the bond between amino acids?
Mm-hmm. And what would that look like?
Like where the COH is on one group and where the NH2 is on the other one?
Yep. Do you remember what functional group that was?
Amide.
Mm-hmm. Yep.
And the COH is carboxylic acid.
Mm-hmm. All right. So yeah, why don't you show me where those amide or peptide bonds are?
But it wouldn't be as OH because it lost the, so
Mhm.
it's just an O.
Okay.
Or wait, no, there's an OH here.
So show me where the peptide bonds are.
Is it this? NHCOOH.
So you said there were amides, right?
Yeah.
So
Oh, that's
yeah.
not amide. Wait, but there is an amide right here.
Mm-hmm.
So this is an amide and this is a carboxylic acid.
Okay. So the amide bonds are peptide bonds. So show me, where else they are. So you have one here, 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 right here, here.
Yeah.
Oops, not there. Here. So okay. So so each peptide bond is going to be between two amino acids. So now what type of amino acids do you see?
I'm assuming the SH is something. There's this CH3,
Mhm.
CH2
Okay.
thing. I don't know what amino acids I see though.
So
Still really struggling with amino acids. Sulfur is in the STY ones. No,
no.
sulfur's in methionine
Okay. Anything else?
and serine.
Sarin is one of the STY ones, though.
Right.
Mhm. So cysteine,
Cysteine and methionine.
yeah. So how do we know if it's cysteine or methionine?
The S. Oh, which one it is.
Mhm.
Isn't cysteine not non-polar? Never mind, it's polar.
Mhm.
We look at the this isn't, it's still double bonded, so this isn't a partial negative, partial positive. Like there's no partial charge.
What, where?
Yeah.
Why isn't there a partial charge?
Because there's still the hydrogen A and B, there's always still a double bond with the oxygen.
So
Does
what?
hydrogen need to come off in order for it to be, does it have to be like O minus, or that would be a full
That would be a full.
um
So we have our cysteines here that can participate in sulfide and disulfide bonds,
yeah.
right? And then we have so another cysteine, another cysteine, another cysteine.
These are all cysteines, not methionine.
Yeah, because methionine,
Has more hydrogens and carbons.
uh, no, the methionine has a sulfur bound to another methyl group, but
Okay.
has the thiol groups. Can you put your phone somewhere?
I don't have my phone near me.
Okay. All right, so we have a bunch of cysteines. What do you think this one is?
E proline? No. That one goes back in. What is that, a phosphate group?
Mhm.
Which ones have a phosphate group? Is it the ones that have the like ring?
So why would those have?
I don't know.
So which are the amino acids that can get phosphorylated?
I don't know.
Um, so they're the ones uh that we talk about usually, STY.
Oh,
Do you remember how they or why they get phosphorylated?
No, I don't remember why. I remember you saying that they do that though.
So what do they look like?
They have the ring structure,
No.
the benzene ring.
Um, so tell me about the amino acids, like what do they stand for?
Three, tryptophan, tyrosine, and serine.
Is there anything else?
Those are the STY ones, but we're talking about
Those are the ones.
charged ones right now, and there's two more or three.
No, no. So STY, what do they look like?
Well, T and Y have the benzene ring.
Nope, they don't.
What do you mean they don't? I have always drawn it. There's always the like thing with the two,
What is the
three,
third like?
huh?
So what do they all have in common? They all must have something in common, right?
Right.
So what do they have in common?
Not sure.
Uh,
Because Sarine's polar and those aren't in the polar group. That was the charged group.
nope, they're all polar. So you could look at this chart here.
How is this not a ring?
Well, you're saying that STY all have rings.
No, I said that tyrosine had a ring.
You said that they all had rings.
Threonine doesn't either.
Sarin doesn't either. And they all have something in common that's not a ring. Like, because when you said they all have rings, like phenylalanine has a ring, but that's not an STY. Tryptophan has two rings, that's not an STY. Histidine has a ring, that's not STY. Uh, Proline has a ring, that's not STY. So it's not that they have rings that make them behave the way that they do. There's something else.
Hydroxyl.
Mhm. And so what can that act as?
A nucleophile.
Okay. So just like they would attack a carbonyl group like this,
Yes.
they would also attack phosphate groups that look like this.
Okay. All right.
See how, so tell me how, like why? Like what did the,
So they're attacking it, but which one is it?
oh no, I'm just asking, so since we know that an OH will attack a carbonyl carbon, right?
Right.
But look at the phosphate.
Because it has the same like structure as a carbonyl carbon
Huh?
with the oxygen double bonded, and then this is like OH, but it loses nodes.
Yep, so STY are going to be the phosphorylatable amino acids because they have that OH group.
Okay.
Okay.
Mm-hmm.
So that's what this one likely is. It's probably serine and threonine. And then what about this one?
So one of these three?
Yep.
No? Or yeah.
So it's arginine, right?
Right, because of the carbon. Yes.
Mm-hmm. And then the other one that I circled.
O and OH, that's one of the more simpler ones, so it has to be one of the first. Or
One of
no, those are just carbons. Never mind.
So what is this functional group?
Carboxylic acid
Mm-hmm. So which amino acids have those as
too.
functional groups? Yep.
But there's two, so it's glutamic acid.
Yeah, I think it might be aspartic because we have one.
Two carbons. Two.
Well,
Oh, but this is the okay, I understand. Yeah, one carbon.
mhm.
All
Okay.
right.
So we could see that. I didn't know what you said, but okay. So now they can also, they also kind of tell us here, like if we weren't, able to recognize them, we can kind of do a little bit of, I guess, logic to figure it out. So they're saying compound one contains 5 regions. There's the hydrophobic region. Where do you think that would be?
Here.
Sure. Uh,
One of the cysteines.
so are cysteines polar or non-polar?
Polar.
So would that make them hydrophobic or hydrophilic?
Hydrophilic.
Mm-hmm.
Right.
So what can be the hydrophobic portion?
But none of them were uncharged. Like non-polar.
I mean, none of these are charged, though.
Right.
So,
But non
mhm.
or negatively charged thing. The aspartic acid is negatively charged.
I mean, not in what we're seeing here.
Okay, so if there's no like negative charge, we still assume that it's non-charged, uncharged.
So, what does it mean to be hydrophobic?
Would be non-polar,
Yep.
but none of these were non-polar.
What about that region?
Is that an amino acid? That's CH3C.
It's just a long carbon chain,
Okay,
and long carbon chains. I mean, carbon compounds, when they're just carbon compounds, are not polar.
okay.
And so that's especially so for a long chain of hydrocarbons.
Okay, so
Okay.
that's non-polar.
That's the
The next one, that's hydrophobic, can be cross-linked for structural integrity. That must be the cysteines because there's a bunch of them.
yeah, and they could be cross-linked, meaning they can form disulfide bonds, right? All right.
Um, spacer residues.
Yep. So those can just be considered, well, since after that they say a residue that was phosphorylated. So,
Though the phosphorylated is this. So it must be this region.
yep. And so these you can see that, well, the R groups aren't being drawn, which would mean that the R group would actually be hydrogen, in which case you would have glycine as the stuff in the spacer residues.
Okay.
And then we have our phosphorylatable residue. And then we have residues with affinity for cells, which can be pretty much these, because these are, I mean, uh, hold on. Sorry, that's someone at the door. Uh, so okay, yeah, these residues at the end, like although they're drawn in a way that does not show that they have charges, in whatever, when you put it in water or something like that, it'll take on these charges. Like for instance, the arginine would gain like a hydrogen here to make it a positive charge because it's basic. And then this aspartic acid here should donate some protons.
Okay.
That's pretty negative. So pretty much this hydrogen can leave to be a negative, and then this hydrogen can add to the arginine to be positive, and that's the region that's going to have an affinity for cells.
That's 5. Okay.
Yeah. So okay, now since we're talking about which amino acid residues were incorporated into compound one.
Adhesion
Mhm.
of cells on the scaffold surfaces. We said those were wait. Is that like the structural integrity part?
Uh,
Scaffold.
well, what are they saying that it?
Promote the adhesion of cells on the scaffold surfaces.
Okay. So where would that be in compound one?
I think it would be where the cysteines are.
Wait, so what are we looking for in the question stem?
Amino acids.
Well, yeah,
Which ones are incorporated?
to do what?
To promote adhesion of cells.
Mhm. What does that mean, adhesion of cells?
That it would bind to the cells.
Yep.
Oh, residues with affinity for cells. This.
Yep.
Okay.
Mhm.
Either aspartic acid or oh, it's D.
Mhm.
Both of them.
Yep.
Perfect. Which amino acid was as a future set of covalent attachment. I want, but I got it right. So it's I'd rather leave it here so it doesn't show me which answer is correct, and I want to try to solve it again because I must have guessed. As a future set of covalent attachment to HA. I don't remember what HA is. Oh, hydroxyapatite.
Mhm.
Hydroxy. I'm not sure.
So it has to be something in compound one, right?
Yes.
So why don't you read where it tells you what compound one consists of?
Hydrophobic residues that can be cross-linked for additional structural integrity.
Wait.
Oh, residue that was phosphorylated for future attachment of HA.
Yep.
The phosphorylated region. I don't remember what we said it was. Hold on.
Yeah.
I can't remember which one was phosphorylated. Like what the amino acid there was.
So which ones must it be or it could be?
STY.
All
And we
right.
said serine. Structurally. So serine.
Okay.
Compound 1 is designed to exhibit pH-dependent self-assembly, which feature of the molecules are responded for pH dependence of aggregation, the hydrophobic region.
Sorry, I keep having someone trying to buzz me for some reason, but okay. So okay, yeah, what is this asking?
It's asking for the exhibit pH-dependent self-assembly, so I assume that it would be something that's charged, so it would be polar, hydrophilic region, which
Okay.
this was non-polar.
That part, yeah.
Yes. I don't know.
So which one of these would respond to a change in pH?
But what would a change in pH do to it? Like, the amino acids are bound to their things.
Okay.
So those wouldn't change, right? Like, can't really denature.
Uh-huh. So maybe we want to look for
Something that isn't bound, really?
no, something that if you change the pH would affect the amino acid. like which amino acids in compound one can change, you know, yeah.
Can change
Yeah. Like I don't mean change into a different amino acid. I mean just change from what we see already written.
this
Uh-huh. And there's the other one too. That one, right? Because you have a basic
oh
arginine and you have an acidic, I think it's aspartic acid. And if you change the pH, you're going to change the charge of those amino acids, right?
hmm
So your answer will be C.
but how is that a side chain?
What do you
Because
mean?
it's on the side.
These are all side chains. This, right?
Because this is the main structure, and so anything that's okay.
Yeah. Like how else can you tell apart amino acids, right? They all have different side chains.
Okay.
Mhm.
Right.
Mhm. Okay. What are your thoughts? Well, what are capillaries?
Capillaries are like they flow to like all the points in the body, but the arteries are larger.
Wait, what does that mean, flow to all?
Main blood vessels.
What do you mean by flow to all parts of the body?
Like the artery is the main like blood
No, no,
supply,
capillary.
and then the capillaries like they're like branches off, and the capillaries flow to like wherever it needs to go.
All right. And then what about like veins?
So capillaries branch off of veins.
So, okay, so let's, I guess, start with arteries. Tell me about arteries.
Arteries are like the big, well, I know that they're the important ones because like if you nick an artery, that's really bad. Like
Okay, why?
because I guess where that's where like the blood supply like around the body, like they're located in the arteries, and the arteries pump it out to the veins and then to the capillaries.
So,
I think of capillaries as like smaller
uh-huh.
than anything else. Like visually, I remember like I've told patients like if they're like, oh, like I have a little thing, and if I scratch it, it's a capillary that like it like causes it to bleed a lot.
So wait, capillaries are where you'd bleed the most from, you're saying?
No, but I'm saying that capillaries are like little things.
All right, can you do, because I know that in like eighth grade and stuff, they teach you like the difference between arteries and veins and capillaries. So do you not
I
remember?
took bio in eighth grade. I don't think that we went over heart.
Well, I mean, you did, you're like a bio major, right? But like
Yeah.
they do teach you as a kid, like what the difference is between the three. So just think of like the circulatory system,
Okay.
but
Veins are
they teach you this like, you know, like middle school and stuff, or at the
most
very least
definitely did
college.
not learn.
What's up?
Most definitely did not learn this in middle school.
No, they definitely. I mean, if not that, then at least like in your bio classes, like the difference between a vein and an artery and a capillary.
I know that arterial blood flow is the strongest
And why would that be?
because It's just how it is.
So imagine asking a doctor that, and they just told you that's how it is.
Okay. Am I thinking like heart system? Arteries are the ones that carry it out. Veins bring it back in, right?
Uh-huh.
Capillaries. I know they're the smallest.
Okay. So maybe they're the ones in between.
So they connect them.
Mm-hmm. So, what's the purpose of circulation?
So that you have like oxygenated blood.
That, but I mean, what's the point of that?
I think you need to have it.
What needs to have it?
Like the like appendages and stuff like that go away from the heart.
Well, so like organ.
Because heart and lungs are like near each other, so
Mhm.
like the heart like has the most oxygenated blood and then it needs to be like sent out to the rest of the body, like to the organs, to the like
So,
appendages
so the arteries are going to be
or
sending oxygen
out.
blood out, and
And veins bring non-oxygen, like oxygen depleted blood back to the heart.
yeah. So what happens in between there?
Capillaries connect them. So I guess
So if we know
like
that,
once the artery sends it there, it enters the capillary and then goes into the vein and then comes back to the heart.
so how do we get oxygenated blood in the arteries to what is it in the veins?
Non-oxygenated. So capillary must be like a membrane that
So,
allows it to pass through the oxygen.
uh-huh. So the capillaries are going to be what lets the oxygen go from the blood to the organs, the tissue, the cells that need it. And that's why they offload their oxygen from the oxygenated blood in the artery through the capillaries. And the capillaries are so thin, they're like the diameter of a blood cell, like one blood cell.
Mm-hmm.
And that means that they have a high surface area, and that one blood cell that can, fit in single file can have gas exchange with the organs or the tissues or what have you, right? Because all the cells are trying to do cellular respiration, right?
Mm-hmm.
And cellular respiration, like meaning like not just glycolysis, but the Krebs cycle and the electron transport chain, right? And all of that requires oxygen, right? And
Yeah.
so all of the cells in our body that are, working, they're undergoing cellular respiration, which means that they're making or that they're using O2 and they're producing as a waste product CO2, right? So when you have like let's say these are your arteries, these are your veins, and then kind of like in between you have these super thin but numerous capillaries. And
Mm-hmm.
so at this capillary level, or rather, let's say this is some organ or some tissue that's going to have, I mean, if it's doing work and stuff, it's going to have um, a low concentration or partial pressure, you can like use them kind of interchangeably here, of O2 and a high amount of CO2, right? And then our arterial blood here is going to have, since it's coming out of the retinol, what? No, coming out of the, like, it does its pulmonary like kind of thing where it goes to the lungs and picks up a lot of O2 and offloads, gives off a lot of CO2. So the blood that's coming in into the artery is going to have, well, high O2 and low CO2, because that's from when you're breathing it, you breathe it in. So what you have happen through diffusion is oxygen travels from the oxygenated blood, which is high in O2, to the organ, because the organ has less O2, right? And then the organ has excess CO2, which it's going to offload to the blood, the oxygenated blood. And so this is all happening. I guess I wrote the arrows to be like kind of from the artery, but they should be from the capillaries. The capillaries is where the gas exchange happens, okay? Now, after this happens, right, the blood will now have a low amount of O2 and a high amount of CO2, right? So the veins will now be carrying deoxygenated blood. And if this is your heart, the veins go in to this right atrium, left ventricle, and the left ventricle just kind of goes, I'm going to draw it like behind, like it goes to the lungs, where now at the lungs are going to take in oxygen from the like outside environment, so the lungs will have a high O2 concentration. And since you're exhaling out the CO2, at the lungs you're going to have a low CO2 concentration, and so that will, I mean, there's going to be capillaries as well, but there will also be, like, after, like in the lungs it'll be capillaries because that's also where gas exchange happens, and it'll oxygenate the blood and get rid of CO2 in the blood. So now you're going to have oxygenated blood again, which is going to go into this left atrium, into the left ventricle, where it then can come out through the aorta and pump that oxygenated blood from that artery back, throughout the body. And same thing, you're going to have gas exchange in your capillaries. So that's the entire thing there. So we can say from this that, hey, what's the original need for all of this to happen, right? The original need for all this to happen is that our cells undergo cellular respiration, which means that our cells use O2 and give off CO2. And gas exchange happens at capillaries. So they'll happen where the organs are here, as well as, I guess, the lungs are organs too, but as well as at the lungs. So cool. Now, the blood is being pumped by the heart, right? And this left ventricle part, right? That's the that's the strongest part of the heart, because that's the part that pumps the blood out from the aorta. The aorta is the largest and strongest artery. And so it pumps it with, so remember how you said like, and it's really bad to nick an artery.
Mm-hmm.
Now, the reason it's really bad to nick an artery is because it's has such high pressure, because it and has such high pressure because the left ventricle pumps it so hard that it lets the blood flow throughout the body. Whereas, it starts to slow down in the capillaries, and then in the veins, it's also slowed down. And that's why veins have valves.
Right, so the one that I did is wrong because it says that the blood pressure in capillaries is the highest.
So let's not worry about that right now. So
Okay.
the veins, right?
Mm-hmm.
That's why the veins have valves.
Okay.
You know what I mean when I say valves? Like, um, kind of
Like,
look like, well, let's say this is the vein and this is a valve. Like, so the purpose of valves,
it's like the channels, right? It keeps it from all flowing in at once.
no, no. So if you see, if this arrow represents blood flow going up, the valves are in this shape. You see how the shape is like kind of like barbs that are kind of like pointing downwards.
Mm-hmm.
So, oh crap, hold on. Wait, sorry. I drew it upside down. So, okay. Again, blood flow this way. Um, the valves will have this type of shape. Now, this type of shape means that it can move up normally, but if it moves backwards, right, these valves will start to lower and be like horizontal like this, preventing the backflow of the.
Mm-hmm.
So, so all in all, we got to think about it this way. Our cells, and we're made up of cells, we use cellular respiration to break down glucose and make energy from it, right? And that
Mm-hmm.
means that we're using up oxygen and we're creating carbon dioxide as a waste product. And so our blood needs to be the thing that transports oxygen throughout our body. And so the arteries that come out of, like I also remember like, and you said this like right, like arteries away, right?
Mm-hmm.
So arteries, since they're going outward from the heart, they have a tremendously high pressure. And then in the capillaries are what are in between the are basically kind of like what connects the arteries to the veins. I mean, technically there's also arterioles and venules, which are kind of like the arteries turn into arterioles and then turn into capillaries and then venules and then veins. Uh,
Mm-hmm.
and so, yeah. So, okay. So that's basically that. So now if you look at the choices here, they're asking why is blood flow slower in capillaries than in arteries? So, okay, let's see, right? So yeah, as you said, the blood pressure is not going to be higher in the capillaries because if they were higher in the capillaries, you'd probably burst them or something, but the arteries will have the highest blood pressure just because that's where the blood has to pump to go into the rest of the body. So yeah, it won't be D. Now, what could, what about these other choices?
So C is wrong
Why is that?
because the cross section is like the area. The area of an artery would be much greater than of a capillary.
They say total.
Total, like across the body, like the number of capillaries.
Mhm.
Mm, there
Like,
would be a lot.
so remember how I said capillaries are like
And
a blood cell, one blood cell, like one red blood cell thick?
Mm-hmm.
Why do you think?
But there'd be a lot of them
Uh-huh. Why
because
would
there's,
they be, you know, one blood cell thick?
I mean, an artery is definitely bigger, but the total, I'm not sure. Like, let's keep it on the back burner and I'll cross out.
No, no,
A.
I'm asking a question here. So why would why do you think capillaries would be one blood cell thick?
Because red blood cells are so numerous. So the smaller,
So so arteries are not one blood cell thick, veins are not one blood cell thick. Well, why would capillaries be one blood cell thick? Like what happens at capillaries?
the channel,
Wait, what?
capillaries are the channel for the arteries to veins
No, but I'm asking why would it be one blood cell thick?
to slow down the blood flow. Like it decreases the,
So if we had something with a ton of these red blood cells, so what when you think of the capillaries, think of gas exchange, okay?
so this is oxygen leaving and CO2 coming back in.
Uh-huh. So if we had this drawing down here, there's an awful lot of red blood cells here. And
Yeah.
that only the ones that are at the ends, closer to the walls, would might be able to have some gas exchange, but very little. Whereas a capillary, if it has a diameter of one blood cell, it forces the blood cells to be in single file, right at the interface of the capillaries. And that's how it can enable, because remember, the gas exchange is happening at the cellular level.
Mm-hmm.
And so you're having, O2 enter in and CO2 come exit out.
So if it's thinner and they have to go like one by one,
Yep.
then it has the time to diffuse
Yep.
out.
Yeah.
Okay.
Yeah. Now, since ultimately, like the volumetric flow must be constant throughout the entire circulatory system, right? Um, and so what that means is the overall, I guess, volume of the circulatory system has to stay constant. And since we know that an artery is significantly thicker than a capillary, since a capillary is as thick as one red blood cell, how
Mm-hmm.
can it do that, right? How can you you're not going to be able to fit in
So you have to have a lot more of them.
exactly. So the total cross-sectional area of the capillaries exceeds that,
Arteries and veins.
yeah, exceeds that of the arteries, um, as well as veins, because that's the only way that it can participate in gas exchange. Uh,
Okay.
so yeah, and you kind of see how I drew more than one capillary here. So
Yeah.
they could be described as a capillary bed. Um, and then
The
B,
area where there's a bunch of them.
mhm,
It's a capillary bed. Okay.
yep. And so yeah, and then B is talking about resistance to blood flow, um, being larger in capillaries than arteries, and the resistance is kind of sort of similar to resistance in circuits, meaning if you have a heart if you have a decrease. How should I put this? So you know how in circuits, resistance is like proportional to length and inversely proportional to area. It's kind of the same here, with that proportion. Like if you have a longer, like whatever blood vessel, it's gonna encounter more resistance. Whereas if you have a higher, sorry, where you have, sorry, compared to if you have a higher area, which would cause a decrease in resistance. So although it's true that we have a higher total cross-sectional area, specifically, this question's saying like just capillaries compared to arteries, not like the total amount of capillaries, so that's not going to be it. And then A says the capillary walls are more elastic than arterial walls. The elasticity is going to be, well, inversely proportional to the
Resistance.
cross-sectional area. Yeah, because the stuff that goes into the capillaries are going to be, like we said, in single file, so it's not going to really matter. Like you're not going to have capillaries vasoconstrict or dilate the way that they would for like an artery or like a vein or something like that. So so yep, pretty much that. So that right there. And it's because of the word total. But yeah, it should also make sense that both blood pressure is highest in the arteries. Yep.
It sounds good. Can't see, sorry. Okay, I got this one wrong too.
Okay. Oh, this is a weird looking thing. Okay. Graph below shows the relationship between the predominant form of iron as a function of solution pH and applied potential. Based on the graph, which of the following statements is true? So x-axis is pH, y-axis is potential. Okay, and then let's see. Let's take a look. Yeah, let's take a look at the choices here. So, oh, you could do, oh, I guess you could do that for the ones that you answered, right?
Yeah.
Okay. Let's see, at a potential of negative 0.4 volts over here, as pH increases going this way, Fe2 plus is reduced and precipitates as, well, okay, what's the problem there?
Because wouldn't it precipitate if it's oxidized?
Well, no,
Like
so,
H2O.
if you just read like the graph,
It's a solid, so it can't precipitate.
no, so what's the graph showing us, and then what's the answer choice showing us?
As pH increases, Fe2 is reduced and becomes Fe3. Oh, Fe. This isn't there anywhere.
Yeah, that's showing FeOH3, but it should be FeOH2, right?
But it's because this one's higher up than Fe2.
So
If it's reduced and go downwards.
so what is answer choice A saying?
At a potential of 4. Okay, so that's here.
Mhm.
Okay, that's the problem. That it was at the potential of 4.
Yeah.
At potential of 4, the Equilibrium between Fe and Fe2 plus is independent of solution pH below pH 6. I don't know what that means though, independent of solution pH. What's the solution pH?
So what's this graph showing us?
Form of iron as a function of solution pH and applied potential.
Okay.
But solution is it this? Fe3 plus?
What are you asking?
Like, I don't understand what the solution pH is.
Well, so like if you look at the graph,
Yeah.
like what does it say?
It doesn't say solution anywhere.
I mean, it doesn't have to say solution, but what
I don't know.
so what are the axes here?
Potential and pH.
Okay. So,
V is voltage.
okay.
I don't know.
So it literally says what it, it's telling us.
But that's pH. I don't understand what that has to do with solution.
It says solution pH.
So that's just the value of the pH value of the solution.
Yeah.
That makes sense. I have no idea. I thought it would be more complicated of an answer.
No. I mean, if it says so look, it says that the graph shows a relationship between the type of iron as a function of solution pH and applied potential. So
Okay, as a function. All right, I understand.
like, because if you're saying that it says pH but it doesn't say solution pH, then why not say this says potential but not applied potential?
Okay, I get it. That it's just assumed. All right.
Yeah, I mean, it's not even really assumed. It just tells you that the graph is a function of solution pH and applied potential. So with graphs, you always want to look at the axes first.
Mm-hmm.
Then you can see. So it's telling us that, it's showing a solution pH and applied potential. So that can only be, one thing, right? The axes.
Yeah. Okay. All right, then is independent of pH? No. Because it's a relationship between, it's definitely not independent. No. pH equals 1.
Why? For B?
Huh?
So why is B not right?
Because if you're saying that it's like the pH of the solution, then it can't be independent because these things start at 0 and go all the way to 6. Like it's not independent of the solution below pH of 6.
Um, so uh how? So okay, if it was at a pH of 4, it'll be here, right?
Right, so that's still like in their cross section.
so if this is your pH of 6 and potential of negative 0.44, negative 0.44 is here.
Mm-hmm.
So
That means that it is independent until it reaches the 0.6.
yeah, right, because it's right on the line that's, touching Fe solid and Fe2 plus. And the change in pH doesn't affect it if it's a pH below 6.
Oh, so independent of the solution, like any value between pH of 1 and 6
Yep.
would keep it the same. Okay.
Yeah. So like
So,
independent means, without it mattering what the
okay.
is, uh like it's below pH of 6.
It would stay the same.
Uh-huh. Yeah, independent of pH.
Okay.
Oh, what about those other choices?
C at pH 1, the potential is changed from -2 to 8. It's the opposite. Goes from 8 down to -2.
Uh-huh. And so,
So C is wrong.
whoops, negative 0.2 to positive 0.8. And yeah, it's just the Fe2 plus doesn't change, so that's out. And then D.
At pH 8 and V 0.1.
Negative 0.1.
FeOH3, not FeOH2.
Yep.
So B.
Hmm.
Octahedral, is that?
Yeah, so that's one of the more complicated bond geometries. And what it really means is something like
Should I close this
um
table?
yeah, you can close it. So this shape here is basically this kind of shape. If you imagine just two pyramid, like take a pyramid and then another pyramid upside down and then put them together like that,
Mhm.
that's going to give you your octahedral, which is called that because it makes a shape with 8 faces. And so we have like, okay, SP, SP2, SP3. So like SP is going to be like a linear structure. SP2 will be like a kind of like a ketone, like a trigonal planar
Mhm.
shape. SP3 is like a regular single bonded, tetrahedral shape. But then once you start introducing the D orbitals, you start to get, and that's with like those transition metals, you can start to get all these other types of bond geometries. Um, and P is, and we're looking for hybridized orbitals, like mixing of like S and P and stuff like that. So P alone won't work. But yeah, you could also hit solution here, and there should be a nice like table here that you can kind of like maybe save and memorize at some point. But yeah, octahedral right here.
Okay.
Mhm.
So octahedral is D configuration.
Yeah, D2sp3.
Okay.
Oh, that previous one was right.
Yeah.
Okay.
You've done a lot with ideal guess.
Mm-hmm. Good job. Uh, okay.
Um, was opsin something to do with like prescription, like opter we talked about, I think. Diopter. So is opsin something similar vision-wise?
Um, let's see. Oh yeah, yeah. Like generally, yeah. It probably comes from Greek for like vision or something like that. But all right, let's see. So um oh yeah, and then this is what's there in our retina. So yeah, vision related. I mean, it's not going to be technically related to like the diopter stuff that we talked about, but it is in the broader category of vision related.
But
Um,
they're different.
mhm. Yep. So okay, human rhodopsin is a GPCR found in rod
Oh,
cell.
you want to read the
Oh yeah, we don't have to. Depending on how, yeah. Let's see. So is this one you want to go over?
Yeah, I got this one wrong.
Okay.
Do you think it's like a high yield thing or
Did you get more than one wrong for this? I'll just kind of like just read it really fast.
two, three?
Oh,
I got the out of wrong.
all right. Yeah, I'll read it. Um, you could hide it again.
Should I read it? Because I
Yeah.
don't remember. Okay.
Go for it.
Human rhodopsin is a G protein coupled receptor, GPCR, found in rod cells of the retina. Rhodopsin is comprised of opsin, a family of 7 helix transmembrane proteins. Oh God. And covalently attached coenzyme retinol. Retinol is derived from retinol, also known as vitamin A. When rhodopsin is exposed to light, the 11-cis retinol, figure 1, converts to all-trans retinol. Subsequent um, structural changes in rhodopsin allow it to interact and activate the intracellular G protein transducin. The signal is turned off via phosphorylation of rhodopsin by rhodopsin kinase, and subsequent binding of the protein arrestin to rhodopsin. Rhodopsin has a maximum absorption of 500 nanometers. Optional opsin-based receptors are expressed in specialized cone cells with maximum absorptions of 420 nanometers, 530 nanometers, and 560 nanometers when reconstituted with 11-cis retinol.
All right. So, what type of amino acids, just because they love asking this question, do you think would be in this transmembrane region? You don't have to tell me specific amino acids. You can tell me what type.
Transmembrane would go through it. So polar?
What does a membrane look like?
There's hydrophobic and hydrophilic.
So,
Like the heads are hydrophobic and the tails are hydrophilic.
what are the,
The opposite.
sorry, tell me that again. So, what are they like? If you were to draw them, like, what would they kind of look like? Cool.
I'm really bad at drawing with the trackpad. I'm sorry.
No, this is really good. So now, uh, so inside is the inside of the cell, right?
Yeah,
And outside is outside of the cell.
yeah.
Now, in the inside of the cell, is there a lot of like inside of the cell? Is it what is that mostly consist of?
Like the membrane,
So
like
we have
liquid,
the membrane, right?
right?
And so the inside of the cell is the cytosol, which is essentially water, like mostly water.
Mhm.
And the outside of the cell is like the interstitial space or whatever, which is also predominantly water. So the heads are actually pointing towards the polar regions, right? Now, what are these made out of, by the way? Like, what is if I took one of those?
Phospholipid.
Good. And what's special about a phospholipid?
They have the polar heads and the non-polar tails.
Yep. So the polar region here, would it be these or these?
The polar region would be these.
The heads, right?
Yeah,
Non-polar region would be in the
inside.
so is this the transmembrane region or this?
The transmembrane region would be. That transmembrane was going from outside to inside, like, and then back out. Like it's going through
Uh-huh.
the inside. So
So
this would be transmembrane region.
good. So what type of amino acids could be present there?
Non-polar.
Good. Good, good, good. Good. So yeah, remember that. And that's also why a steroid hormone with its rings can just go right through the membrane. Doesn't need, to hit a receptor or anything like that. Good. So, because they like, whenever they say this, like, get prepared for a question that's going to be like, what amino acids in the transmembrane region?
Mm-hmm.
Good. Now, what does this indicate about this molecule?
Wait, I was looking at my pen. Sorry. Hold on, I'm closing it. Retinol?
Yeah, with that's this with that ending.
That means it's an alcohol, right?
Nope.
I'm not sure. I remember.
So if it ends in AL, think about things that might have that. Not necessarily at the end, but what functional group it has at least.
Aldehyde?
Yep. And what about this one?
Carboxyl?
Uh, so if
No. Hydroxyl.
yeah, good, cool. So okay, they are saying that this is or rhodopsin is a GPCR that's found in the rod cells of the retina, and that it's composed of opsin, which is a family of these transmembrane proteins, and a covalently attached coenzyme retinal. So retinal, the aldehyde form, is derived from all trans retinol, the alcohol form.
Oh, so then reading through this, I'm rushing ahead. Never mind. Continue.
Oh no.
I figured out the answer. It's C. Because reading this, it was like when exposed to light, it converts to transretinol, which isn't what it needs to go through the processes. So it has to like go back to 11 cis retinol.
uh well, yeah, I don't know about that, but let's not focus on that right now. So, okay. So we're being told that retinal aldehyde form is from retinol alcohol form, right?
Mm-hmm.
And retinol alcohol form is vitamin A, right?
Yeah.
Okay. And then they're telling us that when rhodopsin is exposed to light, the 11 cis retinal, figure 1, converts to all trans retinal.
Yeah, the vitamin A.
What about
Okay. It's vitamin A.
what about it?
All trans retinol, also known as vitamin A.
Oh no, I'm saying that when rhodopsin is exposed to light, the 11 cis retinal converts to the all trans retinal,
Okay.
being aldehyde form, right?
They both. Oh, retinol to retinol. Okay.
Yeah, and then retinal, like 11 cis retinal to all trans retinal. So okay, then subsequent structural changes in rhodopsin allow it to interact with and activate the intracellular G protein transducin. So it can then activate the transducin and then all right, the signal is turned off via phosphorylation of rhodopsin by rhodopsin kinase. And kinases are those transferases that use STY to transfer phosphate groups, right?
Right.
Okay. Um, So yeah, it can be turned off via the phosphorylation of rhodopsin and subsequent binding of the protein arrestin to rhodopsin. Rhodopsin has a maximum absorption at 500 nanometers. What can you tell me about that wavelength of light? Yeah, what can you tell me about that wavelength of light?
Visible.
Good, perfect. Good job. You're killing it. Okay. Additional
Oh,
opsin.
that's colors. Okay.
Additional opsin-based receptors are expressed in specialized cone cells and have a maximum absorptions at 420, 530, and 560 when reconstituted with 11-cis retinol. So it's also present in the cone cells. And the cone cells, there's 3 types that seem to have, peak absorbance or absorptions at these 3 different wavelengths. Okay, and then we see our figure here. And we can see that there's an OH for the retinol that ends in OL. And what's this over here?
That's in aldehyde.
Very good. All right. Let's see if we can answer this without reading further. So,
Production variety of opsins functions too. Why variety of opsins? There aren't.
so
That's when the opsins got phosphorylated.
So, yeah, when we read up, like when we didn't finish reading and you chose something as an answer, that is not the answer. So you would definitely want to like not jump to conclusions like that. And you want to make sure that you, at least in this case,
Opsins are expressed in cone cells. They didn't say anything about A. Doesn't say anything about D. I don't think I read the passage when I answered.
yeah. And then
Like D.
as we went over it again, we read part of it and then you made another answer that's probably wrong. So you want to slow down a bit. So okay, so yeah, we can get rid of D. Keep going.
I think A is wrong.
Keep going.
Enable detection of different colors. Maybe because these are like the wavelengths for different colors, but ensure fast recovery. I guess it never says about it going back to 11-cis retinol. Activates the G protein. So B?
Yeah.
Okay.
But you want to be careful here because so like you chose D at first, and then when we were reading this first paragraph, you skipped over it.
I read this part, but then it never goes back.
Yeah. But that's the thing. If you're just reading this part and then you're choosing your answer, you're not going to get that right answer.
Okay. All right, so
That's
B.
why I crossed it out and then I was like, let's focus.
Okay.
Good. Oh, and then see, I knew they were going to ask this, right? So what do you think the answer will be?
It said non-polar, which is hydrophobic.
Yeah.
D.
And see the transmembrane protein. They always will ask you something about that. All right, now,
Hold on, was this one wrong? Yeah, it was.
yeah,
Yes. phosphorylation of rhodopsin by rhodopsin phosphate group,
You're
right?
sorry,
Huh?
you were breaking up.
Why is my internet connection unstable? Okay, is it better now?
Mhm.
Do you hear me?
Yep. I can hear you. Can
Okay. I was saying, yeah. I was saying that if it's phosphorylated, then it means it's getting rid of the phosphate group, not
Huh?
source of the phosphate group. Signal is turned off via this.
Sorry, what were you saying?
That are added to rhodopsin.
Mhm.
Al, when it's exposed to light, converts to retinol. Subsequent changes allowed to interact and activate this. Okay, so retinol is the source of the phosphate groups. And then when it's turned off, the phosphorylation is removing those phosphate groups back by the kinase. So the kinase does the opposite. But arrestin, I don't remember.
What do you mean when you say it does the opposite?
So I'm saying that phosphorylation is getting rid of the phosphate group, right?
Um, no. If you phosphorylate something, what are you doing?
Adding a phosphate group.
Yep.
How? What is the
So this is a subtle thing with the language that's used. So, um, so it can help. So I really, it can help you to rephrase the question in your own words to understand what it's asking. Um, because I think you're going too fast when you're reading the question stem and not understanding what it's asking. So what is it asking?
where does rhodopsin get the phosphate groups?
Correct. And so,
But rhodopsin is phosphorylated by rhodopsin kinase,
yeah.
which is
And so,
why I picked the answer to begin with.
wait, what?
Which is why I picked that answer to begin with. Because that's what's phosphorylating it.
Yep.
Is there like another thing that acts on it?
Well, are they asking for what phosphorylates it?
The source of the phosphate groups that get phosphorylated.
Uh-huh.
Okay.
And so,
But don't the phosphate groups come when when rhodopsin is exposed to light, the 11-cis retinol converts to Al-trans-retinol and subsequent structural changes, which is the phosphate groups,
nope,
allow it to interact and activate the intracellular G protein, transducin.
nope. That's not what's happening there. But this is where it's happening. And that's why you got to know your stuff about kinases because that's like super high yield, right? So kinases are transferases that use STY to phosphorylate things or dephosphorylate things. It just really is responsible for transferring phosphate groups. But it does it so STY, they'll have your uh hydroxyl nucleophile
Mhm.
and your phosphate would be from, I'm just going to draw the other three like this. This is ATP, right? And this is the alpha phosphate, the beta phosphate, the gamma phosphate. You remember this? And then this attacks here, moves here, comes back, breaks this off. So kinases, you want to know all of this stuff for because it's super high yield. You'll always see something asked about this. So what kinases do is they transfer phosphate groups with the assistance of STY. And if they're phosphorylating something,
Let
right?
me just add it to the note to review for today.
Mhm.
Sorry. All right, continue.
Yeah, so if they are going to phosphorylate something, that doesn't have a phosphate, they can take the gamma phosphate from ATP, which is the outermost phosphate, and they can then transfer that to some substrate, which in this case is rhodopsin.
Okay.
So the enzyme is rhodopsin kinase, that is true. But since they're asking for the source of the phosphates that are added to the rhodopsin, the source will be ATP.
ATP. Okay.
And if you didn't remember that, you can also see that if they're saying the source of phosphate groups,
Adenosine triphosphate.
it's
Okay.
the only thing that has a phosphate. Rhodopsin kinase doesn't have a phosphate. All-trans retinol doesn't have a phosphate.
Forgot.
Doesn't.
Wasn't even mentioned.
Yeah.
Okay. All right.
Oh, it's another one of these things. Okay. Well, at least this one doesn't have the.
Yeah, this one I got right.
All right, cool. Because it makes that trigonal.
I must have flagged. I hate that it doesn't show which ones I flagged. Numbers of carbon atoms involved trans to cis is trans 3 and cis. No, wait, trans is more than 2 and cis is 1.
Wait, what do you mean by that?
What does trans to cis retinol mean? Oh, this to this.
Uh-huh. Yeah, so trans and cis deal with, well, they're going to have a double bond, but if the substituents of the double bond are in the same position, they'll be cis. If they're in the opposite position, they'll be trans.
That's with like the configuration, how this is straight like an alcohol and this is like,
Yeah, so for instance, this compared to this.
So if they're in the same configuration, it's easier like this.
So you want to, when you look at that, you want to think, okay, on the left side of the double bond, um,
What happened to my camera? Do you still see me?
no.
Oh wow, I have a button on my Mac is touchscreen and I accidentally touched the like
So for figuring out trans versus cis, you want to think about left, like what's on the left side of the double bond, what's on the right side. So here, the left side of the double bond has something down, and the right side has something going up. So this is trans. But for this one, the left side is going down, the right side is going down. That's cis, right? And here we can see for the all trans, right? One's going down, one's going up. Whereas here, at least at the 11th position, right? You have cis, right? Because this guy and this guy are both in that same direction.
Yes.
So cis is also like the thing that introduces that kink in olive oil.
What kink in olive oil?
Oh, like you know how like so like
Like
oops.
when you mix it with
Oh, just so like one so like olive oil is liquid at room temperature, whereas butter is solid at room temperature. And the reason is that butter doesn't have a cis bond in it which means that it can be packed closer together, producing a solid. But if you have a cis, like if you imagine this compared to this would be easier to pack together. Whereas, because it makes this kind of like almost straight looking kind of shape, right? Whereas this is making, here, how can I draw it like this? So this looks like that, right? But this looks like that. The cis creates that kink that puts a bend
Okay.
in it. And if you have a bend like this, as opposed to this, the bend is harder to pack closer together. And the example that they give a lot of times back, whatever in some chem class or whatever is like the cis example is olive oil creates that kink, can't be packed in as closely together. So at room temperature, it's liquid, but butter doesn't have that cis bond and can be packed in closer.
Because
So
it's okay.
yeah. Now the thing is, it doesn't matter if it's trans or cis in terms of the shape. The shape, if it has a double bond, like it will be trigonal planar, which is, same thing with like a ketone or an aldehyde, right? Blah blah blah. Whenever you have your double bond, you're gonna have your trigonal planar shape. Like it's flat, and this is 120 degrees between each of them.
Okay.
Mhm.
All right. But what does this have to do with like hybridization?
Oh yeah, so the sp2 looks like these, right? Sp on its own is linear, so that could be like CO2 or like hydrogen cyanide, linear. Sp3 is single bonds. Sorry, I should have said. So Sp would typically involve triple bonds, but in the case of CO2 can involve double bonds, but you're ultimately going to get a 180-degree shape.
Mm-hmm.
Sp2 is going to give you your trigonal planar shape, which is 120 degrees. Sp3, oh, and so Sp2 would be a lot of times just a double bond. Sp3 is going to be single bonds, and that can be like a regular tetrahedral. Um,
Okay.
Something like this with 109.5 degrees. And then when the D ones come in, it can get annoying, and that's where you can use the screenshot that you took of the bond geometries. So, yeah, if we're going from trans to cis, we're still having, our double bond, which is still going to be that trigonal planar shape, which is sp2.
Okay.
Yes.
Like you lost me at some point there, but I think I understand that like this is SP2, SP1 is linear, and then this is one dimensional,
Uh,
was it?
this will be tetrahedral.
SP3?
Mhm.
What does that look like? That's this?
Uh-huh. Yeah.
Okay, all right, sounds good. And then once deconfiguration, that's the like crazy thing. Okay. All right. Catalyzed by what's this? Should we do it? Since I got it right. I don't know.
Yeah, how many did you get wrong for this one?
Oh, this is a different passage?
Yep.
Okay. Two.
You got this one, it seems.
Yeah.
37. Good job.
Okay.
Then let's see how many
One,
here.
one, two, 2 out of 4.
Well, there's more.
Oh wait. 2, 3, 4.
All right. Yeah, let's go over it. All right. Yep. Go for it.
Glycogen synthase catalyzes the lengthening of glycogen polymers through the addition of glucose. The reaction consists of transferring a glycosyl molecule wait, I lost my place. Glycogen polymers through the addition of glucose. Consists of transferring a glycosyl moiety from the uridine diphosphate glucose to the non-reducing end of the glycogen molecule, along with the release of UDP. UDP glucose is formed from the enzymatically catalyzed reaction between glucose 1-phosphate and UTP. It was discovered that approximately 1 out of every 600 to 1500 glucose molecules in a muscle glycogen contains phosphate groups covalently linked to the C2-C3 positions as monoesters. It is hypothesized that glycogen synthase is responsible for the phosphorylation.
All right, we have our structure of UDP glucose here. We have our glucose on the far left, drawn as a chair. We have these two phosphates attached to UD. This entire thing, I guess, is UDP. It's uracil diphosphate. And yeah, I'll say for this that when this happens, this is going to be similar to a little bit similar to how kinases take that gamma phosphate, but it'll be forming a bond with like the second the beta phosphate here. But okay, yeah, and then this is the glycogen synthase, which makes glycogen, which is the polysaccharide form of glucose in our bodies. And they're saying that it adds to this non-reducing end. And remember, that would be like, the acetal or the ketal. Yep. And then it happens with glucose 1 phosphate. And that 1 out of every 600 to 1500 glucose molecules in the muscle glycogen contains phosphate groups covalently linked to the C2 and C3 positions. All right, cool. So I guess for this question, you got C2 position, so C1, C2, C3. And so they said that they get phosphorylated at 2 and 3, so good job. Good job on that.
All right, next one. Type of bond is glucose synthase by glycogen synthase. I always get confused with the alpha 1,4 and alpha 1,6 linkage bonds.
Yep. So if I had
Like I know it's not a beta linkage, but like how do I upon the release of UT?
So this is the 1 position, 2, 3, 4, 5.
No, you're missing. You're missing this one.
Yes. So basically, if we had something one oh damn 1, 2, 3, 4. So if you made a bond between, so 1,4 bond would look like this. I mean, it'll be just a single oxygen. But you could also have, oops, the 1 position and 6 position here form a bond. And that's what will form what's called branches. in glycogen. So glycogen can, so like imagine these circles are molecules of glucose. So these are connected by 1,4 linkages, which make the chain linear. But the 1,6 linkages create branches like that. And ultimately, the purpose of that is to make the overall glycogen molecule more soluble because it has a lot more surface area compared to if it was just a linear chain.
Mm-hmm.
Yeah. And so the linear chain is held together by these like 1,4
Like,
linkages, and the
yeah,
branches are held by the 1,6.
1,6 linkages.
Yeah. And then good. The beta ones are not going to be things that we can humans can break down, but like cows and stuff can do
Ruminants.
exactly. So okay, these are out, and now we just have to think about if it's the yeah 1,4 or 1,6.
Okay, so glycogen synthase upon the release of UDP. So does this, like, is this the UDP
Yeah, this is the UDP.
with the phosphates?
Yep. The U comes from this being uracil. The DP comes from double phosphate, or yeah, two.
Okay, diphosphate. Okay, I understand. So it's forming a bond from this OH, but we don't know that it's forming a bond. How do you know?
Mhm.
Because it could, oh, it's not forming a bond from the 6, it's forming a bond
Mhm.
from here. So that's a 4.
Mhm.
Okay, so it's A.
Mhm.
All right. In addition to glucose, what other monosaccharide is part of the UDP glucose structure?
Oh yeah. So this is the glucose. This is the uracil. These are the diphosphate.
Yeah,
So
so what is
that
this?
is just your ribose sugar. That is
Oh,
the
okay.
building block for like ATP, UDP, uh, any type of DNA or RNA.
Okay. All right. So this structure is ribose.
Yep. If it didn't have this oxygen here or hydroxyl, it'll be deoxyribose.
Right, because it's okay. Right. I got this one right and it was based off of this. So should we skip it?
Um, it makes sense, you're saying?
Yeah.
All right.
Is it the P32? Okay.
Oh, yep. Another radioactive decay problem. So,
Yeah, but I remember I was really confused about this one because I was like, what do you mean, what atoms?
uh-huh. So yeah, so okay, so tell me when you say what do you mean what atoms? Like,
Like it says, it results in the formation of which two atoms?
yep.
And I was really, really confused because I haven't, I didn't know what they were asking.
So we know that radioactive, so what can you tell me about radioactive decay? Like what happens?
It's the same thing as beta decay, right?
Well, beta decay is a type of radioactive,
Radioactive decay.
but what generally happens in
It breaks down the
uh-huh.
glucose.
I mean just in general, radioactive decay, what's that?
it breaks down the molecules into different forms of it. Like,
Yeah, particularly atoms. Uh,
yeah.
and so if it can break it down, they're asking, what would it form?
So this one, glycogen synthase. I have no idea. Like,
So what are the radioactive stuff that
C, P. Is it these?
when you say these, what are you?
DPUC and U and beta P.
Uh-huh. So these are the radioactive like labels.
Uh-huh.
Okay. And so if these undergo radioactive decay, it's asking what would they form? Because if an atom undergoes radioactive decay, it changes that identity of that atom, right?
Right. So it's carbon and phosphorus.
As the radioactive ones. Yep.
Yeah. Oh, and it says C14. So that's N and P32. Where's P? Here, 32 is S. So is it N and S?
So tell me, like,
It's going under a conformational change and it's like it's basically an ion, right?
nope. Uh, so a conformational change is when something, changes shape or something.
Okay, sorry, wrong word.
Mhm.
So it's going the gamma, the radioactive decay
Uh-huh.
that's happening is converting it. So it says we're either C14 or P32 radioactive decay.
Mhm.
So if this happens as described, it would result in the formation of which?
Yeah. So what type of radioactive process is happening?
Like in general or in this problem, it's beta decay.
All right.
Okay,
So that, right? So,
right.
uh, think about how you would, write out the beta decay.
I don't know how to write them out.
Okay. So let's try. So you can hide the solution if it lets you.
Because I didn't pick one.
All right. So okay. So like C14 and carbon would have a atomic number of 6, right?
Yes.
And if it breaks apart if it undergoes beta minus decay, the beta particle has a 0 mass and -1 charge, right?
Y is a -1. What's your God? Oops.
It says beta minus decay.
Oh, okay. Yeah, so 0. But why is it 0 and not if the original is 12?
Oh, well, a beta particle has a mass of 0.
Okay.
So what would this turn into?
-1 charge.
Yeah.
Huh?
What's that?
Saying it would like -1 charge, but the 14 stays the same. I see 14 here.
So what do you
-1 would be -7. I don't know what.
so everything on the left, like the numbers on the left have to equal the numbers on the right.
So 14 and 5.
Yep. Wait, are you sure about the 5?
Wouldn't 6 -1 be 5?
Why would you do 6 minus 1?
Plus -1.
Uh-huh. So everything on the left has to equal everything on the right. Right?
Right.
So for this new, atom that we're writing here, what must its atomic number be?
6.
So that would give you
Oh, 7, because then -1, it would give you 6,
good.
which is the exact thing for n. Okay, all right, I understand.
Now this one,
32 beta 0 -1, so it would be S. Remember that, and then 16. 32S16. Yeah.
so
Okay.
there you go. So yeah, I would yeah, maybe take a screenshot or whatever of this and study your you're getting
Kinases.
a lot better with your functional groups, very good. Um but review.
I know, I really want us to go over BB.
Oh, I mean, I think that was like a pretty good score. I
I
think
know, but I feel like there were conceptual things that I like wanted us to go over when I was doing it.
uh-huh. Yeah.
We continue going over, like I take the exam and we like, so I don't have lessons scheduled for next week, I realized. I guess I stopped on Saturday. I don't even have one scheduled for Sunday.
Yeah. So you should put
Yeah,
the
I went on Calendly and there's no days like for this week. You're like completely booked out.
um yeah, that's
Like
why I
it
always say
was,
to make those sessions, but I see some availabilities. Like I see
so on Monday, it's like Monday and Tuesday, it's when I'm in class. Like I have class until 3
uh-huh.
and yes, I can't do either of those times.
What about like the rest of the week?
The rest of the week just says booked out.
No, I mean
What, an hour.
what is your availability for the rest of the week?
Show you my schedule. The thing is I have these 2 lab days that those I can't miss my labs. Like if it's a lecture class, I can skip it. But you see, wait. Yeah, this is next week. So you have Monday was at 1 p.m. I have genetics at that time, and then Tuesday was at 3 p.m. and I have lab.
Hmm.
But and then these days I don't see at all on the thing. I have your Calendly open somewhere. Maybe I was doing it on my phone. But Yeah, I didn't have.
Hmm.
Here I'm in class until, I don't know why it's not here, but I have a class like this. Or no, sorry.
Uh, so what times work for
I
you?
can do like any day after 5
Hmm.
or in the mornings. Like I don't have classes until later. Whenever you can squeeze me in.
Um, yeah.
Like I don't see anything for the week. If
Yeah.
I select an hour, look at it. There's like Wednesday. It's completely done. And then Thursday, I leave.
Hmm.
Thursday I can do if you have time, but I just have a my preview that day.
Yeah. Um, okay. I'll try to like maybe message some of my students to see if I can move them around, but
Really sorry, I should have done it. I didn't realize. I like completely forgot that I only scheduled it because I did it like 2 weeks ago. I
yeah,
know you told me to go all throughout, and then
I
you
was
never.
yeah, that's why I always said that you have to schedule in advance.
Yeah, I'm sorry. I know it's just like the last 3 lessons.
Yeah. So,
But
yeah, I'm not sure. Um, yeah, I'll see if I can move some people around, but uh, I don't know
should I book the times, like the Monday time for now?
which time.
The 1:30 on Monday.
Um, I mean, is that something you can do or
If I miss that class, then yeah, but that's not a lab class. So that one's like more or less like it's not great, but I can miss it.
uh-huh.
Lab, it's like a whole ordeal if I miss.
Yeah. Uh, yeah, I'll see if I can move some people around, but I
All
think
right.
you can also try to see if you can fit anything in, like if
Well,
like or like
Tuesday, I definitely can't do that 3 p.m. one, but
uh and then
Monday, I just scheduled for 1:30.
what about Wednesday and Thursday?
Wednesday is blocked off.
No, I mean like what would you be like
I'm
what
good with anything. Wednesday and Thursday, I can do whatever times you have. Wednesday, I'm done at like, she usually lets us out by 4, so 4:30 I can do already.
okay. So yeah, I'll see what I can do here.
Okay, and then Thursday I have like no classes, so I
Mhm.
can do the whole day. I have like my preview at
Okay.
night.
All right.
Uh,
We're meeting tomorrow though, right? 6 p.m.
yes, yes, of course.
And Sunday you're gonna take a test.
Yes. Yeah, I'll take it on Sunday. Perfect. Since we're not meeting on Sunday.
All right then. So all right, then I'll see you tomorrow. Okay?
Okay. All right.
All right.
Thank you so much.
See ya.
Bye, Shin.